Okay, good afternoon. Thank you very much to the organizers for inviting me. I'm really happy to be here. As John mentioned, I work at IRRI in the Philippines on drought physiology, and one of the big parts of my job is to get genotypes from the drought breeding group, which we know are doing well under drought in terms of yield. And my role is to try to understand what are the physiological mechanisms that are conferring that yield advantage. So I'm going to focus on root traits today. So I thought I would start with just giving a little bit of background about rice roots, because just so that we're all on the same kind of terminology. So this is a rice root seedling. Actually, I don't know how to use this. Do I press a button?
Turning, there's a little.
Okay, thanks. Okay, this is a root system of a rice seedling, and there are three main types of roots. So these coarse diameter roots are called nodal roots. The fine diameter roots are called lateral roots, and this long one is a seminal root. And later on in the growing cycle, these nodal roots will become longer than the seminal root. And even though these lateral roots look really fine in diameter, but actually they're not called root hairs. You can't see any root hairs in this image. But if you take one nodal root and magnify it, then you can see root hairs. And basically root hairs are single-celled, and they're very small diameter. Usually you need to use a microscope to see them. And another thing I would like to point out about rice roots that's very unique is the anatomy. So compared to other crop plants, rice roots have a couple of unique features. One is a predominant amount of parenchyma. So these are cortical cells that have died, and they make air spaces. Because rice is adapted to flooded conditions, those air spaces help get oxygen to the growing root. Another important anatomical adaptation of rice is this sclerenchyma layer that's usually suberized, and it's thought to help reduce radial loss of oxygen to the rhizosphere under flooded conditions. So this is a wind rhizo image that we've already been introduced to wind rhizo by Steve. But I just wanted to point out about rice roots. There are actually several different classes of lateral roots, each with distinct diameters. And you can see that the red roots that are colored red are called S-type lateral roots. They have a very small diameter, less than 0.05 millimeters in diameter. So they're really fine. And there's also L-type lateral roots, and this big coarse one is the nodal root. And as was mentioned earlier, this software allows us to quantify the total root length within the specific diameter classes. So those are some of the tools. This is one of the tools I'm using. And actually, IRRI has had a lot of research, root drought research going on since the mid-1970s. So here's just some old images. This is from IRRI annual report where they're showing comparing two genotypes, IR20 and OS4, where OS4 had, this is a density diagram demonstrating deeper root growth in what was characterized as a drought-resistant genotype. This is a photo from the 80s where they were using aeroponics to characterize root depth as well as diameter. And this is some work from Brigitte Courtois looking at root distribution with depth. And during all of this research, really the main focus was what they called deep and thick roots. So they were really looking at nodal roots mostly. How deep were they growing, and how large was the diameter, because they thought that was very important for penetrating compacted soil. And just to put this into context of the drought breeding program in general at IRRI, throughout the 1970s and 80s, there was a lot of screening going on, both in the greenhouse for root traits and other traits, and also in the field for drought resistance. And their strategy was really screening for survival. So they would give a really severe stress and then rewater and score for what were the genotypes that were surviving. But that actually, survival is not really correlated with yield. It was later realized. So then later, during the 1990s, there was a lot of focus put on breeding for traits, so selecting for specific traits where the physiologist would recommend a trait and then the breeding group would try to integrate that trait, particularly deep and thick roots. And during that time, they were able to identify QTLs for root traits and integrate those root traits into different backgrounds. However, that strategy also, they were able to integrate these QTLs, introgress QTLs into different backgrounds, but they couldn't make the connection between having that root QTL and having better yield under drought. So then, in about 10 years ago, they made a really dramatic shift in their strategy. They decided, okay, no more traits. We will just select for yield under drought. And that's not that easy because you don't get a good drought treatment if it rains, and you have to have a very uniform field so that it drains uniformly. You don't have too much air in your experiment. But after working for about a decade on this, they were able to identify major effect drought yield QTLs. And I think this is a pretty unique situation in rice. I know in other crops it's not really a common thing to have major effect QTL for yield under drought. And this is a slide from the breeding group showing the different QTLs they've identified, additive effect, the donor, and basically these are all QTLs or segments of a chromosome that were identified for reproductive stage drought stress, and they are introgressed in the background of popular mega varieties. So these are varieties that are grown on millions of hectares in target environments in Asia, and they're high yielding when it's well-watered conditions, but they're very drought sensitive if there's not a lot of water. And the two that I'm going to be focusing on talking about physiology are QTL 12.1, which is from a Weiraram Vandana cross, and also a set of QTLs from Adesel in the IR64 background. And interestingly, some of these QTLs, they've been identified from multiple genetic donors, and they are effective in multiple backgrounds. So I'm really lucky to have such nice genetic material to work with because they're very similar to the parent genetically, but they have such dramatic differences under yield. However, I still have a lot of challenges at IRRI, namely the weather. So doing drought research at IRRI can be challenging. This is the bulletin board I see when I come to work. Sometimes it's kind of depressing, today, tomorrow, day after, rain, rain, rain. But during the past few years, we've been able to make some progress in terms of our infrastructure. So we've got 4 new rainout shelters that are automated, so they close automatically when it starts raining. And we have a sensor rack we're working on that has can measure canopy temperature and NDVI, and we're working on a couple of other different sensors. And we've also been we've set up this lysimeter facility in the greenhouse. So basically, this is a way of keeping track of water uptake rates by weight. And every time we use a mechanized hoist that helps speed things up, and every time we weigh the plant, we simultaneously take a digital image of the shoot so that we can estimate leaf area, and then we can normalize water uptake rates for plant size because, of course, bigger plants take up more water. So that's a couple of the facilities we have, and also I just wanted to give a little bit of background about how we're doing our root sampling. So basically the most common method we use is just by soil coring. So we have a 4 centimeter diameter steel tube that's 60 centimeters tall, and we pound it into the soil. Then we use a chain block to pull the core out of the soil. That step helps make things a lot more efficient. We split up the soil into different segments so that we can get root length distribution with depth. And we carefully wash the roots, trying to keep as many of those fine roots as possible. And then we scan using WinRhizo. And then just to give you a little bit more background about, so rice is different. There's many different kind of agroecosystems. We're working with both upland rice and lowland rice. So in upland rice, it's direct seeded into the soil. Lowland rice is transplanted, and it's usually kept under flooded conditions. This is what these two situations look like under drought stress. One of the biggest differences is that lowland soil tends to crack a lot, and the stress can be pretty severe. But upland stress in general comes a lot faster than under lowland conditions. So of the two QTLs I'm going to talk about today, QTL 12.1 is for upland conditions, and the IR64 odd cell QTLs are for lowland conditions. And our sequence for doing this kind of work is step 1, we get plus and minus QTL lines for breeders that show contrasting yield under drought. Then we put a lot of effort into ensuring the uniformity in our stress treatment and characterizing what kind of stress we have. And basically, we're really focused on doing reproductive stage drought stress because reproductive stage causes the greatest yield losses. But this is a really important step because there's so many different types of drought. Seedling stage, vegetative stage, reproductive stage, moderate, mild, severe. For that reason, we should really know what kind of drought is occurring, not just drought or well-watered. And then finally, we start working on phenotyping. And our strategy is to go from using high throughput methods, then get more and more detailed depending on the direction that our initial results are pointing to. So for example, if the QTL lines are showing better water uptake when the soil is dry, then we look for root-related mechanisms. So, okay, for QTL 12.1, this was characterized previously by having a 7% greater water uptake in plus QTL lines under drought in lysimeters. That was all that was known up to recently. And roots were measured, but the researchers weren't able to identify any genotypic differences. The only thing they could see was differences in water uptake in lysimeters. So one of the first things we did was to measure root growth in the lysimeters. And the results were pretty interesting. So this graph shows soil depth on the y-axis and root length density, which is centimeters of root per centimeters cubed soil. And what we saw was no differences at all in terms of total root length at any depth that we sampled over 2 seasons. However, we did see differences in average root diameter. So that, if you remember the WinRhizo scan, it shows all the fine roots and the coarse roots all together. If you average the diameter of all of those roots, that's what this number is. So we were seeing some differences in average root diameter. So then we went to look more detail into root length within the different diameter classes. So each color is a different diameter class here. And you can see that as on a percentage of the total root length basis, the plus QTL lines were having significantly greater root length in the small diameter classes, but less root length in the large diameter classes. So that kind of explains why we weren't seeing differences in total root length, but we saw differences in diameter, and it's because the diameter classes were kind of shifted. So these plus QTL lines were having basically more lateral root growth, but less of the coarser roots. And we also did a greenhouse seedling stage study, and we saw greater branching in the plus QTL lines. So our hypothesis was that, well, maybe this greater root branching is allowing the plus QTL lines to access the depleting soil water and then conferring a yield advantage that way. So we wanted to look more into the kind of water relations. Oh, but first, we were interested to know why wasn't it that we were able to pick up the differences in root growth earlier? And we went back to the methods that were used. So this is an earlier root scanner. This is called a ComAir scanner. And this is the WinRhizo scanner that we used. And just as we were doing this work, a paper came out by Yoichiro Kato from University of Tokyo, where they actually compared these two scanners, and they saw that this scanner has a very reduced ability to detect these very fine roots. So that was interesting. I think there was a technological limitation. But another thing is that we really have put a lot of effort into using a very thorough washing method. So in the past at IRRI, the method for washing roots was to put the soil sample on a screen and then run water over it until the soil washed away. But if you do that, then there's a high chance of losing a lot of the fine roots. So what we do is we mix the soil with water and let the roots float below the surface, and then we pour that mixture off over and over again onto a sieve. And I think that helps us recover more roots. So going back to the water relations, we did a number of measurements in our rainout shelters, and they show some evidence for a role of QTL 12.1 in water uptake. But you'll see there's still kind of a little bit of doubt here. So for example, for plus QTL lines, we saw lower canopy temperatures. So basically lower canopy temperature when the soil is dry means that somehow those plants are able to continue taking up water so that they don't close their stomates and they're transpiring and cooling the leaves. So the plus QTL lines had lower canopy temperature, but they were lower than the susceptible parent and minus QTL lines, but not lower than the Vandana, which is the recipient parent. Same with soil moisture. So one of the plus QTL lines was drying out the soil a lot more than others, implying improved water uptake, but the other plus QTL line wasn't. And yeah, so it was not quite straightforward. But we did see some interesting relationships between canopy temperature and stomatal conductance. But and then at this point, we wanted to look a little bit more into what was going on. And also, because we're seeing this effect on fine roots, which actually we only saw under drought stress. We didn't see that under well-watered conditions. I wanted to look at different types of soil. So would this greater lateral root growth have a different effect under different types of soil? So we did an experiment in our lysimeter facility, and we used two different treatments, well, two different drought treatments. One where we just used clay soil from IRRI, the other where we mixed sand in with the soil. And the results were pretty interesting. So these graphs show water uptake rate, so grams per day normalized per leaf area. And the plus QTL lines are in blue, and the parents are the orange and green. And when there was no sand, it was interesting. The plus QTL lines actually had lower water uptake rates. And then when there was sand, early on in stress, the plus QTL lines had lower water uptake rates. But by the end, 3 of the plus QTL lines had greater water uptake rates. And this was really interesting, especially because we hadn't seen this kind of relationship at all in our lysimeter facility. So up to now, we've done a few different lysimeter studies. All of them have been under lowland conditions. So basically, the pots start out flooded, then we drain them and let them dry out. We did this with the Oryza SNP panel, which is 20 diverse genotypes, and also with some African glaberrima lines. And in both cases, what we saw was that the genotypes that had greatest water uptake rates were consistently the genotypes with greatest water uptake rates throughout the study. So there was no really crossing like we were seeing in the previous slide. And I think that this kind of makes sense because in lowland conditions, the soil dries so slowly. In the field, after we start our drought stress treatment, usually it's about 2 weeks before we see any kind of drought symptoms at all. So maybe that slow dry down gives the plant time to respond, for example, in terms of root growth. Because in all of these cases, we saw very good relationships between root growth at depth in the lysimeter and water uptake rate. However, in upland conditions where drought happens much faster, maybe the plants need a faster mechanism to deal with that kind of stress. For example, regulation of transpiration. So here, maybe these plants are being conservative by regulating their transpiration, and then by the end of the study, they have more water available and can have greater water uptake rates. But I also thought this was really interesting because this was the first time we saw a result that really correlated well with some other water uptake lysimeter studies. So this is a graph from a paper at IRRI64 in the group of Vincent Valdez, where they see really similar trend, where the drought-tolerant genotype, this is in chickpea, starts out having a lower water uptake rate at the beginning of stress, and by the end, it can have a higher water uptake rate at the end of the study. So yeah, this was interesting, points to the differences in adaptation to lowland and upland types of drought stress. And I also think it'll be really interesting because we have so many different partners that are looking at these genotypes. Mostly we're focused in India, Bangladesh, and Nepal. And during the past year, I've spent a lot of time characterizing the soil at our different partner sites. And basically, I've taken soil samples with all of our partners, and we've gotten them analyzed at the same lab, at the IRRI India lab. And you can see this is a diagram about soil types. So you've got percent clay, percent silt, percent sand. And you can see what a range of different soil types we have. So I think that it's going to be really interesting to look at the yield response in these QTL lines at all of these different sites, especially based on the interesting water uptake results we got. And at a couple of sites, we've even worked on capacity building for root sampling. So now they have all the same equipment that we have for soil coring. They even have WinRhizo scanners. So hopefully at a couple of the sites, we can get root data also. So for the QTL 12.1 part, in summary, QTL 12.1 appears to increase water uptake when the soil is dry through increased lateral root growth under drought, but we also think that other traits or mechanisms can be conferred by QTL 12.1, perhaps transpiration efficiency. So that's something that we're working on characterizing in more detail now. So next, I'll move to the QTLs that are in the lowland background. So this is ADECEL IR64 lines, and this is one that a lot of people are excited about this year. Actually, both of these lines are now in multi-location trials in India, and we're trying to release them as varieties. But this was from our rainout shelter this season. You can see IR64. This is the recipient background. Again, very high yielding under well-watered conditions, but very drought susceptible. And this is the this is one of the QTL lines. Actually, this one just has 2 QTLs. But you can see what a big difference there is in terms of yield under drought. And this is just a slide from one of our partner sites in Hazaribagh, India, conceptualizing the yield differences that they saw. So this was a trial a few years ago in our rainout shelter. Actually, you can see water here because it was taken just after we rewatered. But this is the plus QTL line, and this is the minus QTL line. So I think it's pretty clear. You can see that this one is having more leaf drying, and this one looks greener. This is an infrared image. So basically, red and white are warmer temperatures. Blue is cooler temperatures. Here's the minus QTL line. Here's the plus QTL line. So that was very dramatic. And it was interesting because I had been growing these lines for a few seasons, but I didn't see any differences, so I wasn't even really interested in this experiment. But then we put it in the rainout shelter, and we were able to get severe stress, and then that's when we were able to see differences. So yeah, this is IR64, the recipient parent. So when I saw these results, I was really sure, okay, these lines, the soil is really dry, but they're still transpiring, so they must have deeper roots. And already we had seen some interesting results relating deep root growth with canopy temperature, where genotypes that had more roots at depth were able to maintain a lower canopy temperature. And again, this was in the ORIZA SNP panel, the 20 very diverse genotypes. So then we took a bunch of root samples from those IR64 ADECEL nils, and surprisingly, we didn't see any differences in root length density at depth at all. This graph shows root length density at 30 to 45 centimeters and 45 to 60 centimeters. So at this point, I was a little bit puzzled because, that I really expected there to be deeper roots. So I thought, well, okay, if we're not seeing differences in root architecture among these genotypes, there must be difference in root function. And rice is really typically known as not being very good at drying out the soil compared to other genotypes. It just has kind of a lower water uptake ability per length of root. So in terms of characterizing root function, we did some studies on root hydraulic conductance using a pressure chamber. So basically, we grew the plants in these soil-filled tubes that were just the size of our pressure chamber. So then we put the tube in the pressure chamber, seal it all up, we cut off the leaves, and then we apply pressure to the root system, and we quantify how much sap comes out at different pressures. So then we can calculate root hydraulic conductance. And of course, at this point, I expected, well, the plus QTL lines must have better root hydraulic conductance because they're able to keep transpiring. But we saw the opposite. And in fact, yeah, the plus QTL lines had significantly lower root hydraulic conductance than the minus QTL lines and the recipient parent. And yeah, so I thought that was interesting. But then we wanted to know a little bit more, like why would these lines have lower root hydraulic conductance? So we started working on some root anatomy. And in fact, we saw that the plus QTL lines had smaller root diameters and significantly smaller xylem vessel diameters. And interestingly, we only saw this under drought stress. This was not a phenotype we saw under well-watered conditions, or not even really under mild drought, only when we gave the plants severe drought. And this was really interesting because actually this was a trait that was published back in the early 80s. Richards and Paciora published some work on the importance of small xylem vessel diameters in wheat. And they were hypothesizing, you can comment on this later, but that the small xylem vessel diameters are restricting water uptake and maybe a mechanism for saving water for later. So that's one possibility. Another possibility might be that these smaller xylem vessel diameters could result in decreased xylem vessel cavitation. So when there's really high evapotranspiration demand, maybe the smaller vessels are less likely to cavitate. But at the same time that we were doing this work, interestingly, we were just trying to do some kind of basic characterization of root anatomy and root hydraulic conductance in response to drought. And we were looking at a few genotypes, Dular, IR64, KDML. So the top and bottom ones are considered more drought resistant than the middle one. And in this situation also, we saw that the more drought resistant genotypes were decreasing their xylem vessel diameters when the stress became more severe. Whereas IR64 didn't, I mean, there was some decrease there, but not as significant as in the two more drought-resistant genotypes. And we've also been looking at this bleeding rate in the field. So basically, it's where you just cut off the whole shoot, and then we collect the sap that comes out. And we compared a number of different genotypes, including Dular, IR64, KDML, and Swarna. And what we saw was that Dular and KDML, the drought-resistant genotypes, consistently had less sap coming out than the drought-susceptible genotypes and the high-yielding genotypes. So Swarna and IR64, high-yielding genotypes, are up here, and Dular and KDML had lower bleeding rates. So finally, we went back this season, and I just wanted to check what was going on with these IR64 Ade cell nils. And in the line that was performing best, indeed, it also had lower bleeding rates under drought stress. So it just kind of validates what we were seeing in the pressure chambers and also with the root anatomy. But there's still a lot we don't know. I think, yeah, it's been interesting to see significant differences, but I can't say we can really explain all of it. So one thing is, I would really like to do more to understand how would smaller root and xylem vessel diameters under drought result in improved yield. Also, I mentioned that in this picture, this line only has 2 QTLs, whereas originally they had identified 4 QTLs in that set. So we want to do more on understanding the effects of individual QTLs and different combinations. And we want to investigate the leaf-related mechanisms of these nils, because so far I've really been focusing on roots a lot, not so much on leaves. So in summary, I think these results highlight that root traits can indeed contribute to improved rice yield under drought. We're not seeing the same kind of phenotype that was originally hypothesized at IRRI, which was deep and thick roots. And I don't want to say that I don't think deep and thick roots can be effective. It's still a possibility, it's just that that's not what we were seeing behind these specific QTLs. Another point is that because a QTL is a part of the chromosome, it's not a single gene. So just like it might not be single genes responsible for that QTL, it also is likely that it's not single traits behind that QTL, right? So for example, with QTL 12.1, we think maybe there's some kind of water uptake rate mechanisms going on, and we're still investigating more with the IR64 Ade cell nils. And then finally, kind of a philosophical statement, I think that this approach of looking at yield first and then going back to traits, for me, it's been really enlightening, because I think it allows us to be open-minded in understanding mechanisms for drought resistance. Because, for example, if we are just recommending traits to breeders, then we're limited by what we think in our own minds could be effective. But kind of going this way, it allows us to get genotypes that are already showing significant differences in yield under drought, and then we can try to figure out what's different from that perspective. So next, as I mentioned, we're doing a lot on soil characterization, and we want to look at different effects of the environment. And we also think it would be interesting to pyramid the QTLs. So maybe these QTLs have pyramided traits in themselves, but then it would be interesting to see if some of these traits can be complementary or synergistic when we put them together. And finally, we just came out with a manual, a small manual on methodologies for root drought studies. And I've brought one for all the students in the course, and I have a couple extra if anyone else is interested. But it could be helpful for anyone that wants to start doing some root work. And this is our group. We have lots of technicians to do root scanning. And I would like to thank especially the drought breeding group. This is Arvind Kumar, the drought breeder at IRRI. They've given us some really nice genotypes to work with, and they keep us on our toes in all our trials. And our funding is from GCP, Gates, and BMZ. Thank you.